Tonight's lecture covers Chapter 12, the Anatomy and Physiology of Nervous Tissue. The purpose of this chapter is to understand how the nervous system helps to keep controlled conditions within limits that maintain health and homeostasis. Tonight we'll also learn about the different branches of the nervous system, and we will identify and describe the various types of cells that are found in nervous tissue. So starting with an overview of the nervous system, page 420, 12.1 on page 421. And you can see there that we have a cartoon that kind of shows the two different nervous systems that you have in your body, which is the CNS, which stands for Central Nervous System, and that includes the brain and the spinal cord. And then the PNS, which stands for the peripheral nervous system, and then you can see that includes the cranial nerves, the spinal nerves, the enteric plexuses and small intestine, and sensory receptors in the skin. Just wanted to draw the parallel between the nervous system and the skeletal system at this point, because remember, the skeletal system, we had the axial skeletal system, and that included like the skull and the spinal cord, and then the appendicular skeletal system, which includes the arms and the legs and the things that branch off of the center of the body. So it's kind of the same thing here. We've got the CNS that relate to the brain and spinal cord, and then the PNS, which relates to the other parts that are not associated with the brain and spinal cord. So this is still the same figure here on page 421, figure 12.1. And we can kind of see this little flow diagram that has to do with how the nervous system works. So you can see here that there's some sensory input that is either the somatic senses or the special senses, which we'll get into later in the chapter. And they're going to give information to the central nervous system, and then that's going to create what we call a motor output. And the motor output then will feed into the somatic nervous system, which is the skeletal muscle. So it might be if you're playing baseball and somebody hits you a line drive and you move your glove over to catch it, that would be an example of how the motor output in the somatic nervous system would work. The autonomic nervous system, however, deals with what we call the sympathetic, the parasympathetic, and the enteric plexuses. And so sympathetic and the parasympathetic deal with our involuntary muscles. So you see here smooth muscles, cardiac muscle, and then glands. So glands is something that we haven't talked about this semester, or if we have, it's only been in passing. And then the enteric plexuses deals with smooth muscle and the glands of the digestive system, or the canal. The functions of the nervous system. This is still on page 420. Sensory. We detect changes through sensory receptors. Integrative. We analyze incoming sensory information, store some aspects, and make decisions regarding appropriate behaviors. And then motor, which is to respond to stimuli via effectors. And I'll define effectors as the lecture goes on. So first we need to understand the histology of the nervous tissue, and this is on page 422. And you should probably already know this, but histology just means the study of cells. So now we're going to talk kind of about how nervous cells are made up and what comprises those. So we see here on page 423, figure 12.12, this type of a cell is called a neuron. So it's not your typical cell, right? It has a lot of other features that you might not see in a red blood cell or a white blood cell, or remember when we were talking about the cells that make up the skeletal muscles? Looks a little bit different, doesn't it? Well, let's see here. Neurons are electrically excitable, so that means that there's some electricity that will associate with these. I just want to point out, though, that all cells, they have some kind of membrane potential. Cellular structures, so we can see that there's all kinds of structures on here. We've got the dendrites, we've got the axon, we've got the axon terminal and the synaptic end bulb. And then the nerve impulse is called the action potential. Now we'll get into the physiology behind this later on in the lecture, so I'm not going to go in too much detail about action potential at the moment, but just know that when I'm talking about potential, because we're going to later on tonight, we're going to talk about graded potential and action potential. And all potential really means is that there's some kind of potential energy. In the case of the neuron, when we talk about the potential energy, we're really talking about electrical energy. This is part of the same figure, 12.2 on page 423, but here we can see a light microscope image of a neuron. So we see here that the neuron does have a nucleus. All right, and then it's got this thing called an axon. And then it's got these dendrites. So let's go back here. So you see here we've got the dendrites, we've got the nucleus, the axon. So this picture here, this cartoon, is a little more detailed than this, but this is pretty good for a light microscope. And then we see here a scanning electron microscope of a similar image. And you can kind of see that the neuron is a little bit asymmetrical, and it has all kinds of branching points that we would refer to as dendrites, and then it's got this axon and these axon terminals, and this is what we will study tonight. By the time we're done with this lecture, you should understand how the nerves, the neurons actually work, and how, if I was playing baseball and got a line drive hit to me, how my senses can tell me, to move my glove over here and catch the ball. So let's go through some structural classification of neurons. This is figure 12.3 on page 424. Neurons can be classified based on the number of processes extending from the cell body. Let's have a closer look at this. So we've got something that's called a multipolar neuron, and this neuron is probably the most common neuron that we'll run across in our studies here. But you can see here that you got the dendrites and the cell body and the axon terminal. So this is kind of the classic model of a neuron. And then neurons can also look like this, where they've got this dendrite and then this axon and then these axon terminals. But we call this one a bipolar neuron because, well, look what's going on here. We've got this dendrite that is this one stem, if you will, going up, and it almost looks like a tree, so it branches off. Otherwise, it kind of looks the same, right? So you see it just doesn't have all these finger-like projections coming off of the cell body. Instead, I like to call these like tree-like projections because it really reminds me of a tree. Oh, and then I should also point out this arrow. So this arrow really just means the direction of travel of the action potential, which we'll talk about later. Then we've got the pseudo unipolar neuron, where we see now the cell body is located kind of more in the center of the cell, and the axon is actually branching off of the cell body and leading us to either dendrites or the axon terminal. And then this is another detail, figure 12.4 on page 425. But this is just examples of how the dendrites can branch. So the neurons they're highly branched. Not only are they in a kind of your more traditional type of neuron that we see here at letter A, how they're branched with these more finger-like projections. But we can see here that the stimuli is going to move in this direction. So this is what we call a Purkinje cell, and this is what we call a pyramidal cell. I guess both kind of work the same, but they just look different. And we can see here that all of the action potentials, or the charge of the cell, is moving toward the cell body. Once it reaches the cell body, it's going to be directed out towards the axon terminals, where it reaches either another neuron, some kind of neurotransmitter. So these things are going to either articulate or connect to some kind of another tissue or another neuron, where it's going to send the signal on to the CNS and then back to create a motor response. And we'll talk about all that as the lecture progresses. The functional classification of neurons. Neurons can be classified based on the direction of nerve impulse propagation. One is the sensory or afferent neurons. This conveys information to the CNS. The motor or the efferent neurons, which conveys action potential from the CNS, and the interneurons or the association neurons, and they process sensory information and elicit motor response. So let's go through each one of these and talk about what this means. So afferent, I guess that's really the word that I want you to focus on, is afferent. So afferent means that I've received some stimuli, like for instance, I'm a baseball player and playing shortstop, and the batter hit a line drive to me. that I'm getting some sensory information telling me that there's a line drive coming right at me. Where should I move my glove to catch it? Or maybe I need to run over a few steps to catch it. And so there's going to be some sensory information that's going into the central nervous system. So when a signal from an external stimuli reaches your eyes and it tells you what you can do, or it doesn't even have to reach your eyes. If somebody is walking behind you and they accidentally bump into you, well, you can't see that coming, but you certainly feel it. And that's going to give you some stimuli that's going to go into the central nervous system, and that's afferent. So afferent travels towards the central nervous system. And then as a result of that sensory input, we get what we call a motor effect or an efferent neuron. So your brain is going to process its information that's been collected by your neurons, sent to the central nervous system. It's going to process it out, and it's going to create some sort of a motor effect or an efferent. So information traveling from the central nervous system back out to the skeletal muscles is telling you, hey, you need to move your glove up here to catch the ball, or somebody bumped into you, you better move your feet like this so you don't trip and fall. So that's the idea here. Afferent is to the central nervous system, efferent is out from the central nervous system. And then interneurons, I guess it's like your electrical system at your house. If you go to your electrical system in your house, maybe you have a basement or a closet that has a fuse box. I can go to that fuse box and I can shut off the fuse or the circuit breaker, whatever you got. Let's use circuit breakers, for example. I can shut off a circuit breaker to my living room, and then all the electricity on whatever circuit is going to be shut off there. So it's kind of like you got this electrical source coming in to your electrical box, and you've got all these circuit breakers, and as long as they're on, you can get potential or electricity going to whatever outlet or light switch or whatever you got. But if I shut one of those off, there's not going to be any electricity there. So that's kind of like an interneuron. Maybe even a better example would be like if you're using power strips to plug in your TV and your stereo. If I unplug that power strip, then that would be kind of like an interneuron, because you've got the electrical outlet on the wall. I could plug the lamp in directly, or if I plug it into the power strip, then that would kind of be like an interneuron. So interneurons are just mostly associated with the central nervous system, and they mostly deal with helping the afferent signals to come in to be processed and sent to certain parts of the brain. And then that's going to come back out as the efferent, and they're going to go through some intermediate neurons that will help to get that signal to where it needs to be. If somebody bumps into me, I may be getting signal on my backside, but it's telling my brain that my feet need to move so I don't trip and fall. So I guess the analogy I'm trying to draw is that the interneurons are more like just kind of like a junction box that help get the signal to where it needs to go. So let's have a little closer look here at figure 12.5 on page 426. So we see here, this is our peripheral nervous system where we get some sensory input. So this is going to create a nerve impulse, which is afferent because it's traveling towards the central nervous system. It's going to hook up with an interneuron, and that's going to help the nerve impulse to continue to travel. You're going to get some signals from your brain telling you what to do. It's going to connect then with this efferent neuron, which efferent effector, you get cause and effect. Neuroglia is on page 427. So there's different kinds of cells that make up the central nervous system, and one of them is neuroglia. The reason that it's called neuroglia is because at some point in history, they didn't know that neuroglia were electrically excited. They thought it was just kind of what held the nervous system together, but now we know it's much more than that. So we can see here that the neuroglia is not electrically excitable. It makes up about half the volume of the nervous system. They can multiply and divide, so like your cells, they can go through mitosis, and there's 6 kinds. There's 4 in the central nervous system, and there's 2 in the peripheral nervous system. Page 427, figure 12.6. Let's look at the scanning electron first. So we're looking here, we've got this neuroglia cell, which it looks kind of like a cell body, right? And then it's got all these branches coming off of it. And then we see here in the cartoon that when we dig into this cell body, we have all kinds of different little aspects to it. So we see here is the neuron, and we should already be familiar with the neuron because we've been talking about it for the last several slides, but the neuron is going to take in that sensory information, and it's going to send some kind of signal down this axon, and that's going to go out to other neurons, and it could even go to the central nervous system, and that's going to create some sort of reaction. And then you can see here we're going to be talking about these here coming up. So the oligodendrocyte, the astrocyte. So this is another part of the central nervous system that we'll talk about here in a few slides. So the neuroglia of the PNS, basically the Schwann cells, what they do is they wrap around the axon, and if the Schwann cell is wrapped around an axon, that creates what we call a myelin sheath. This is going to be important. So Schwann cells basically create myelin, and if there is an axon that is completely surrounded by myelin, then that axon is said to be myelinated, as it says here. This is really just the part of the Schwann cell. And then furthermore, these unmyelated, so it's kind of hard to see in this view. It might be easier to see in your book. This is figure 12.7 on page 428. But if we look really close here, you can see that this axon is not completely surrounded by myelin. See, there's a little gap here, there's a little gap here. There's gaps around all of these. To me, it almost looks like an old school dial telephone, but it doesn't necessarily have to look like this. There could be several axons like in this area here. But these axons are called unmyelinated because they are not completely engulfed or surrounded or wrapped around with the Schwann cell that creates the myelin sheath. So this is important because the axons that are myelated, actually the nerve impulse will travel faster through a myelinated axon than it will an unmyelinated axon. And we got a figure later that will show you that. And then quickly over here, we see that when we have a group of nervous cells together, we call that a ganglion. And those ganglions are usually surrounded by what we call satellite cells. So you can see here the satellite cells are kind of here on the top and then here underneath it. And the myelin sheath is produced by Schwann cells. We already established that. And the oligodendrite cells in the CNS, and it surrounds the axons of most neurons. So I guess the point is that most of the nervous cells in your body are myelinated. This is figure 12.8 on page 429. So you can see here we've got the Schwann cell, we've got a nucleus. So this axon right here is what I would say is not myelinated. Well, this is kind of a magnified version of that. So the Schwann cell is not completely surrounding that. And furthermore, I want to point out that the Schwann cells, they actually wrap around the axon, kind of like if you've ever done electrical work, how you would wrap a piece of electrical tape around a wire, or, well, hopefully if you're putting two wires together, you're using wire nuts or something. But still, even when I use wire nuts, so a wire nut is just basically a nut, and in the electrical world, there's different gauges of wire. If I'm working with like number 6 wire, I'd get a number 6 wire nut. I twist that wire together, I'd put a wire nut on it, and that gives it a secure connection. And then I would still wrap that up in electrical tape just as a precaution. But if you've ever wrapped something in tape or electrical tape, like a wire, that's kind of the idea of how a Schwann cell is wrapped around the axon. See how it's kind of swirled? It's layered, or whatever you want to call it. I guess this one's just showing us more of the unmyelinated cells. So here's some micrographs of the neurons. And we can see here that this myelinated axon has this thick myelin sheath that is created by this Schwann cell. And as opposed to on this letter D, it's lacking this myelin sheath, but we still have the Schwann cell. And then we can see here that there's very thin, if any, Schwann cell going around this part of the cell, or this, I guess this would be an axon. So this brings us to figure 12.9 on page 30. So the takeaway from this is that the brain is composed of gray and white matter. White matter seems like it's a little more predominant in the brain. This is the spinal cord too. But my point here is that the parts that make up the white matter are myelinated, and the parts that make up the gray matter, which we see here in the spinal cord and in the brain, those are unmyelinated. Page 430, electrical signals in neurons. Excitable cells communicate with each other via action potentials and graded potentials. I already mentioned those two terms, so make sure you're making a note of those. Action potentials allow communication over short and long distances, whereas graded potentials allow communication over short distances only. Production of action potentials or graded potentials depends upon the existence of a resting membrane potential and the existence of certain ion channels. Well, resting membrane potential and ion channels are important because we're going to be talking about those in details here in a few slides, but you should already be familiar with ion channels from Biology 100 or 111. Most cell membranes do have some sort of an ion channel that allow either a passive or active diffusion. So basic biology stuff, I'm not going to go into detail covering about that stuff tonight. You should already know it. Figure 12.10, page 431. I'm not a big fan of this slide. It's really broad brush. I guess there's a little bit of detail in it, but idea is that it's kind of showing you how graded and action potentials work, and it's kind of showing you how peripheral nervous system can take a signal to the central nervous system, and that's going to come back. So, you know, if I pick up a pen, hey, so I'm getting some sensory input from my pen where my fingers are grasping that, and it's sending this action potential down this long neuron, reaching the central nervous system, where it's going to interface with interneurons and go into the brain. And the brain's going to tell me, write the letter A, or I'm going to write a letter to my pen pal across the world. I'm really amazed at how fast this stuff works, because I should be able, you know, I can write pretty fast. I can type fast too. I can write cursive and I can write manuscript. And how does my brain know which one I need to write? And how does it know I got a pen in my hand? And furthermore, I find that when I do write letters to my pen pal, sometimes I'm writing in manuscript, and then sometimes I'm writing in cursive, and my handwriting is kind of a blend between the two. This figure here is just kind of a broad brush of how the signals are taken from the sensory input to the brain and back. So we've been kind of getting into this a little bit, touching on the surface here. Ion channels and neurons, here on page 433, figure 1211. Leak channels alternate between open and closed. Potassium ion channels are more numerous than sodium ion channels, and there should not be a space in between the N and the A. So this is basic biology here, basic chemistry. You should be familiar with sodium, potassium. You should know some stuff from the periodic table, which that's where that comes from. But we can see here that we've got this extracellular fluid. We've got a plasma membrane. And then we've got cytosol. Look at what's going on here. Very simplified diagram is that you've got golden pyramids, or whatever you want to call them. And there's lower concentration of these potassium ions in the extracellular fluid as there is in the cytosol. And then you've got this protein here, or this channel, and that's going to lead to passive diffusion. Yeah, this is basic biology stuff. So you know that what happens then is that a molecule will passively diffuse down its concentration gradient. So since the concentration of potassium ions is higher here in the cytosol, these are going to diffuse down the concentration gradient and out into the extracellular fluid. And that's one of the ways that we create action potential. So we'll get more details about this here in a few. Ion channels and neurons continued. Ligand-gated channels respond to chemical stimuli. So All that a ligand is some kind of a chemical signal that's going to bind to this particular protein that's embedded in the cell membrane. So don't get confused here. It's not an enzyme, but if you remember from 111 and 100, what we talked about concerning enzymes, that it works like an enzyme. Once this chemical or this ligand comes in and binds to this protein, it causes a conformational change, and that protein opens up and it allows some sort of diffusion. And here we can see we got the potassium, so now we've got purple and green. The green is the sodium ions, and then the purple is the calcium ions. So you can see here that those ions are going to diffuse in or out of this particular protein that's been opened up by this ligand. And in the case of this example, it's showing you acetylcholine. We talked about that during the muscular system because it's an important chemical that associates the nervous system with the muscular system. Here's another type of an ion channel, and these are mechanically gated ion channels, so they're going to respond to some sort of mechanical stimuli, whether it's vibration or pressure. Vibration or pressure can open up this channel, and that's going to cause a reaction there where there's going to be some sort of diffusion of ions that way. If you're on the CTA and if you're on the orange line and somebody behind you trips and bumps into you, that's some pressure that, it's going to change that, and it's going to send that signal because that mechanically gated channel has been opened up, and it's allowed you to create some sort of action potential there to send the signal to your brain and move your feet however you need to move so you don't get tripped or fall. Let's see here. So we got voltage gated channels, which respond to direct changes in the membrane potential. If your membrane potential when you're at rest is at negative 70 millivolts, if that was to change to negative 50 millivolts, that would create some sort of an action potential where this protein can open up and allow some sort of diffusion of some ions such as a potassium ion. This is table 12.1 on page 434. So here it just gives you a description of the different types of channels and where they're located. So you can study that on your own. Let's talk about resting potential of the membrane, and this starts on page 434. The membrane of a non-conducting neuron is positive outside and negative inside. This is determined by unequal distribution of ions across the plasma membrane and the selective permeability of the neuron's membrane to sodium ions and potassium ions. Most anions can't leave the cell, and there are also sodium-potassium pumps associated with this. So let me first say that an ion is just a negatively charged particle, and a cation is a positively charged particle. So let's see what this is about. So here we have figure 12.12, also on page 434. And you notice here, if we look at the plasma membrane right here, we see that in the extracellular fluid we have a positive charge, and in the cytosol, we have a negative charge. In this particular model, or cartoon, whatever you want to call it, we see that there are an equal number of positive and negative charges in the extracellular fluid. Right here, we have like equilibrium, right? And when we have equilibrium, if we were to put a voltmeter on that, we would see that our membrane potential then is at negative 70 millivolts. Moving on to figure 12.13 on page 435. So we see here that we have a number of ions, both in the extracellular fluid and in the cytosol. We have all of these proteins that are embedded in the cell membrane. So there's more potassium that will leak in and out of the cell than there is sodium. So it's more common, but sodium can also leak in and out of the cell. And then sometimes we use facilitated diffusion, where we use a molecule of ATP to help pump in either sodium or potassium, depending on what the concentration gradient is at any given particular time. Let's talk a little bit about graded potentials. So graded potentials are a little bit different than the action potential that we were just kind of describing. So we see here that the graded potential on page 436 at figure 12.14, there are small deviations in the resting membrane potential. So small deviations, so that means there's not much stimuli there. And we can see we're here at this negative 70 millivolts, and we've got this resting potential. Some sort of stimuli comes in, and it raises the membrane potential at this time frame, so about 10 milliseconds. and then that's going to cause some kind of a change in the behavior of the cell. And this brings us to figure 12.15 on page 437. A graded potential occurs in response to the opening of a mechanically gated or ligand-gated ion channel. So these deal with gated channels here. We see that we've got some resting membrane potential, and then there's some applied pressure here. So somebody on the orange line tripped and bumped into me, and I got some signal. It mechanically opened some of my proteins, and I was allowed to diffuse in some sort of ions, creating what we call depolarizing, which we'll talk about in the upcoming slides. And when something is depolarized, that really just means that the resting potential has been changed, and that creates what we call an action potential, which will send a signal to your brain and back. Yeah, that's a mechanical stimulus. Let's see here. So we got ligand stimulus, which could be the binding of some sort of a chemical to this protein embedded in the membrane, which is going to open it up and allow depolarizing to occur. Because remember, all depolarizing is we're changing the potential of the membrane. We're creating some kind of a charge. We're right here at this resting potential. Chemical comes in, binds to the protein, opens it up. Ions rush in and out, and that changes the potential of the cell, creating a reaction that will be sent to your central nervous system and back to your periphery, if that's what the case may be. And then hyperpolarizing. Let's hold the thought on hyperpolarizing, because when we get to 1218, I'm going to explain that better, because there's a better slide for that. But let's go along with the PowerPoint in the book. So we're at 12.16, and that's on page 438. But we see the amplitude of the graded potential depends on the stimulus strength. So if that person on the orange line bumps into me and I get stimulated to move my feet so quick that he bumped into me with some force, changes my resting membrane potential, causes a reaction. But maybe he just kind of brushed into me, and that does just, hey, excuse me, you know. Hopefully the guy's not being malicious and trying to pick my pocket. But if he bumps into me hard, hopefully I'm going to, hey, sorry, man, I didn't mean to do that. Or if he just kind of brushes me, you know, hey, excuse me. But we can see here that if the strength is greater than it is, you're going to get more of a reaction than you are to this little reaction. But as you'll see, we also have to get to what we call threshold. So this is considering if we get to threshold. And how much above threshold we go is going to dictate to us, you know, our response. Talk about threshold in a few slides, I promise you. I know it's so exciting. So in graded potentials, though, if we have more than one stimuli, those stimuli can become combined, and that can create more of an enhanced response. So graded potentials can be added together to become larger in amplitude. And all amplitude is how high this wave is going. If I was to have a couple of different stimuli coming at me and it was a graded potential, I could add those together. I could get more amplitude, as we see here. See, so this stimuli is just here. This stimuli is just here. But if I combine the two together, I can go up this high to get more of a reaction that way. There is a point where you actually top out on your potential there, but we'll do that later. And then we'll go back to resting potential here. So action potentials. 12.6 on page 438. An action potential is a sequence of rapidly occurring events that decrease and eventually reverse the membrane potential and eventually restore it to the resting state. Action potentials have two phases, depolarization and repolarization. So we were already talking about depolarization. Depolarization is just we've taken that resting membrane and we've depolarized it. So we've changed its polarity. If we've got a negative charge on the inside of the cell membrane in the cytosol, and we have a positive charge in the extracellular fluid on the outside of the cell, when we depolarize it, we change that polarity. And then repolarization gets us back to our resting potential. This is the diagram I wanted to show you to talk more in detail about the hyperpolarizing phase, or hyperpolarization, as we mentioned a few slides ago. And this is figure 12.18 on page 439. So we've already talked about all this stuff kind of. So we get some sort of a stimuli, and that's going to change this potential. And if we reach this threshold, then we get a reaction, which is going to tell our nerve that, hey, we need to make a move or we need to do something. So we get depolarized, and then we repolarize. And then oftentimes what happens is that after repolarizing and you go back to resting membrane potential, you're going to overshoot that. And I got a great analogy for that. If you ever play baseball or softball, and let's say that you're up to bat, this pitcher's good, and I swing and I hit the ball, man, I don't knock it out of the park to get a home run, but I knock it past the shortstop and it goes out between the left fielder and the shortstop and I get a base hit. But it's a close play because those guys on the other team are fast and they get to the ball and they throw it to first base while I'm running the first base. And I'm running so fast to first base that when you get to first base, you can actually overrun first base. Now remember, when you overrun first base, you want to turn towards the dugout. You don't want to turn towards center field because technically if the first baseman's got the ball, he can tag you out. It just reminds me of this hyperpolarizing, because then once I hit the ball and I get this depolarizing and I'm running, and as I'm repolarizing, I get to first base, I'm allowed to overshoot first base, turn around and go back to first base, or turn around and just go back to resting potential here. That's my big analogy. That's why I love this chapter, because I get to talk about baseball. So let's see here what's going on here. Page 440, figure 12.19. Action potentials can occur only if the membrane potential reaches threshold. So remember, I've already mentioned the word threshold. So before, let me back up 1 second here. Action potentials are what we call an all or nothing reaction. In other words, you're not going to get that nerve impulse unless you go over a threshold. So you've got to get at least a -5 millivolts in your stimuli. You can go higher than that, but not going to get a reaction. In order for you to get some sort of stimulus to move forward with whatever you need to do, that action potential has to be over what we call threshold, which is just kind of the baseline for that. If it doesn't go over threshold, you don't get any reaction. Figure 1220 on page 441 is on one page, but this is actually 4 slides here. So you can see here we're at a resting state. We've got a closed gate here. So this is our sodium channel, and we've got our potassium channel. And we have a positive cell membrane out on the extracellular fluid side, and then the cytosol side, we're negatively charged. But we've got a higher concentration of sodium ions out here, and a higher concentration of potassium ions out here. And then we get some sort of a stimuli where we depolarize. And now all of a sudden these channels open up and we get an influx of sodium ions into the cell, because this is just diffusion, right? Higher concentration of ions are going to move to a lower concentration of ions. It's just a basic law of nature. And we see here depolarization. See this occurring over time. And when we talk about time in millivolts, we're talking about milliseconds. And then as we get to the repolarizing phase, the potassium ions are going to start to move in. And then finally, we get back to where we're at here with the repolarization continuing. Now our concentrations are a little bit more like they were when they started out on the resting state on the first slide regarding this. And for more information on this, you can look at the table on page 444. 12.2. Propagation of action potentials, page 440. In order for communication to occur from one body part to another, action potentials must travel from where they arise at the trigger zone to the axon terminals. This traveling is called propagation. Action potentials do not die out. They keep their strength as they spread across the membrane of a neuron. So there's 2 types of propagation that you should be familiar with. One is continuous, and we've kind of been talking about that. And continuous propagation occurs in what we call the non-myelinated axons. We've got this time scale here where we've got milliseconds. And then we can also see versus the saltatory propagation, how the impulse is propagated through myelin sheaths. And look at the distance it travels in 5 milliseconds, 10 milliseconds. The saltatory conduction occurs much faster than the continuous conduction does on the non-myelinated. It's still fast, even with the non-myelinated cells. It's still going to be a fast reaction, just not as fast. So there are some factors that affect the propagation speed. One is axon diameter. The larger the diameter, the faster the propagation, the amount of myelination. So myelinated cells are going to propagate much faster than non-myelinated. And then temperature. Higher temperature increases the speed of propagation. Page 444, signal transmission at synapses. A synapse is a junction between neurons or between a neuron and an effector. So what I mean by an effector would be a skeletal muscle. Synapse is going to occur at two places. It's going to occur between neuron and neuron, which is very common. And it's also going to occur between, like, for instance, neuron and a skeletal muscle or a gland. You know, maybe you need to release a hormone or something. Like your adrenaline glands, you know, are situated on top of your kidneys. So you have to get a signal to go down and kick in your adrenaline. Electrical synapse. So there's a gap junction that connects cells that allow the transfer of information to synchronize to the activity of a group of cells. And then chemical synapse is a one-way transfer of information from a presynaptic neuron to a postsynaptic neuron. Talk about those two terms in a minute here. So this shows synapses between neurons. This is 12.22 on page 445. So the presynaptic neuron is the cell that has the signal before, so once it passes on to this other cell, that's why it's called postsynaptic, because we've got presynaptic and postsynaptic. And if this was going to pass on the signal to another cell, then this would then be presynaptic to that cell. Right? So presynaptic and postsynaptic depends on the direction of travel of the signal. So here's the signal transmission at a chemical synapse. And this is figure 12.23 on page 446. So you can see here that we've got the synaptic bulb and we've got these neurotransmitters that are located in here. So what happens is that this nerve impulse is going to come down. So now we're dealing with calcium ions. So this is also voltage gated channel here, so there's some electric potential going on. But we've got this thing called the synaptic cleft. So in between this presynaptic neuron and this postsynaptic neuron, there's a gap there, and this electrical signal cannot jump this gap. So what happens is that this electrical signal is going to trigger a chemical reaction, and it's going to release these neurotransmitters that are going to go into these proteins embedded in the cell membrane on the postsynaptic cell, stimulate those, and it's going to allow in sodium ions. Again, just getting back to what we were explaining about resting potential and depolarization and that. So you get this chemical signal that goes into these, they act as ligands, and they bind to these proteins, open up the channel, ions diffuse in, and that's how the signal is propagated in the chemical synapse. So that's a chemical, not an electrical synapse. An electrical synapse is way faster than a chemical synapse. But chemicals, don't get me wrong, chemical synapse is still fast. But since there's that synaptic cleft, we have to have a way to bridge the gap, and we do that with a chemical, what your book calls a neurotransmitter. Postsynaptic potentials, page 447. But it's also, I'm looking at the book, page 446, it's a bold-faced word. So that means it's a vocab word. Something important for you to know. A depolarizing postsynaptic potential inhibits postsynaptic, that doesn't even make any sense, excitatory postsynaptic potentials. So let's see if we can figure this out then. What is depolarizing? Well, that's when the membrane potential changes. The cytosol becomes positive and the extracellular fluid becomes negative. That's the example of depolarization. Hyperpolarizing. Remember hyperpolarizing? I use the example of the person running to first base. And, you know, when you go to first base, if you're just getting a base hit, you go to first base and then you can overrun the base. You don't have to stop on it. Now, second base is a different story. If I'm running from first to second, I have to stop on it. But that's why I like to use the example of first base for hyperpolarizing, because I don't have to stop on it, but I can turn and I can go back to first base and wait for the next batter to hit me into home. So a postsynaptic neuron can receive many signals at once. Yeah, I'm not real keen on that slide. Let's talk about the structure of neurotransmitter receptors. And this is also on page 447. Neurotransmitters at chemical synapses cause either an excitatory or inhibitory graded potential. Neurotransmitter receptors have two structures: ionotropic receptors, which contain neurotransmitter binding sites and ion channels, and metabotropic receptors, which contain neurotransmitter binding sites and is coupled to a separate ion channel by a G protein. So let's look at ionotropic receptors, and this is page 448, figure 12.34. So what's going on with these? You see we're at resting potential here. We see on either side of the cell membrane, we've got a negative charge and we've got a positive charge. We've got some ligand coming in and binding to the protein, opening it up. And in this case, we're using acetylcholine. And then the acetylcholine receptor contains an open cation channel. Remember what I said about cations. You should know the difference between a cation and an anion. Anion is negatively charged, cation is positively charged. And that creates an excitatory postsynaptic potential, EPSP. I mean, that's an example of ionotropic receptors. An ionotropic receptor is a type of ligand-gated channel. We already covered those, but now we're really defining them in even greater detail. So when we have some kind of channel that's ligand-gated, we know that that's ionotropic. On the other hand, we have the metabotropic receptors, and that's also on page 448 at figure 12.24. So once again, we see that our potential here, we're at resting potential. And then we've got this protein that is going to be stimulated by this acetylcholine to move off of this channel. And it's going to come in contact with this potassium channel and open it up. And then it's going to inhibit this potential. So it's going to slow down the reaction. So inhibitory postsynaptic potential is IPSP. Removal of neurotransmitters. So when you're in the synaptic cleft and you have these neurotransmitters transmitting a signal from one cell to another, at some point maybe the stimulus stops and then the signal has to stop. So what do we do to remove the neurotransmitters from the synaptic cleft and to stop the signal? Well, there's three ways that we can do that. One is diffusion. In other words, the neurotransmitter is going to diffuse into either of the cells that are around there, any of the cells that are around there, I should say. Enzymatic degradation, in other words, there's an enzyme that will come in and degrade those neurotransmitters, or they will be uptaken into cells, whether it's the presynaptic cell or the postsynaptic cell. We're getting to figure 12.25 on page 449. If several presynaptic end bulbs release their neurotransmitter at about the same time, the combined effect may generate a nerve impulse due to summation. Summation may be spatial or temporal. Remember, we talked about summation before. What slide was it? Let me go back. Yeah, it says it right here. The graded potential, it's summation, right? So when we have more than one stimuli coming in, we've got all these stimuli exciting our action potentials and our graded potentials. Those are what we call summational, and that can be in space-time, spatial, and temporal. I just said that. Spatial summation, figure 12.25, page 449. So we see we've got these presynaptic neurons, two different ones with some 1 and 2. They both have some stimuli coming into this postsynaptic neuron. So remember, all these neurons in your body, they're making their way to the central nervous system. So it's almost like the superhighway, right? You're coming up I-57 to get on 9094, and I'm coming on 55 to get on 9094, and eventually our cars are going to be on that same road because we're both sending, it's like a presynaptic signal that we're sending to this postsynaptic neuron. We're creating more action potential there because now there's more stimuli on 9094 since both your car and my car are on there. Get it? It's kind of like that, right? So we see that we reach this threshold level, and since you and I got on the road at the same time, it creates this spatial action potential where our stimulus is going to spike. I don't know, like walking and chewing gum, right? I mean, it may sound funny. Well, can you walk and chew gum? You know, I mean, but really, just think about like the synapses that go on there. I mean, you're walking down the road and you're chewing a piece of. So there's all kinds of stuff, and not to mention all of the autonomic stuff that we're not even talking about, like breathing and heartbeats and all. So there's a lot of stuff going on. I mean, these nerve cells they're just amazing because there's so much constant depolarization and repolarization, and it happens so fast, and all these signals they get all over. And if I'm playing bass, I can catch the ball, or you know, I can catch the ball and I know the guy's coming up, so I can tag him out and I get a double play, right? Temporal summation. So all that happens here is that at the same time we get this summational time in milliseconds, and then all of a sudden we're here at the same time getting this action potential. And that's the same figure, right? Hold on, let me check the figure. Yeah, same figure, 12.25 on page 449, 12.26 on page 450. So we see here we've got our EPSPs and our IPSPs. So we've got some excitatory activity going on. We've got some inhibitory activities going on. And then that's just going to trigger, so all this stuff at the same time and space, it triggers this zone of the postsynaptic neuron where it tells your body what to do because of all the stimuli you're getting in at the same time. And then this is table 12.3 on page 451. Summary of neuronal structure and function. So this is just kind of an overview of what we've been talking about tonight. You can see we've got the dendrites, the cell body, the axon hillock. So the axon hillock is actually this part here that kind of bridges the gap between the cell body and the axon, and then the axon terminals. Let's talk about neurotransmitters, and neurotransmitters starts on page 451, but these are small molecules, and it shows examples of what your body can use as a neurotransmitter. Acetylcholine, we've mentioned that before. Amino acids, so you should know amino acids by now, right? Biogenic amines, ATP and other purines, nitric oxide, and carbon monoxide. Those can all function as neurotransmitters. And then here it just kind of shows you the chemical structure of those. 12.27 on page 452. That's what I was saying, like the purines, adenosine, like ATP, adenosine triphosphate. And you can see all these amines. You probably even heard of some of these: norepinephrine, epinephrine, dopamine, serotonin, amino acids. So this you should be familiar with from Bio 100 and Bio 111. I don't know, I taught 100 recently, and they used to have that square codon chart, but now they changed it into like a wheel. So if you've taken that class recently, instead of being a square, it's now a circle. But remember, there's 20 amino acids, and then, you know, those are what make up your messenger RNA. Comes out into the cytosol and gets to the ribosome. Then these amino acids are going to, based on the triplet patterns that you got, it's going to, tRNA is going to affix these different amino acids together, and they're going to make a string, which ultimately becomes a protein. So that's the idea behind the amino acid. Nitric oxide, yeah, this is important for men's sexual arousal. Carbon monoxide is when you turn on your car exhaust creates carbon monoxide. And the acetylcholine, we've already talked about that quite a bit tonight, and in the muscular system, so you should be becoming familiar with that as well. But this figure here just shows all their chemical structures. So neuropeptides, well, this is still figure 12.27 on page 452. I think I'm going to skip this. So this is a better description of that because it actually tells you what the substance is and what it does. So I'll let you go through this table on your own. By the way, that table's 12.4 on page 454. Neural circuits, page 454. A neural circuit is a functional group of neurons that processes specific types of information, types of circuits. Simple series, diverging, converging. So diverging means they go apart. Converging means they come together, reverberating, and parallel after discharge. So here's an example of diverging and converging neural circuits. So you see we've got some input, and see how they divide or they diverge. So you get 2 of these neurons stimulated, and then 4 of them are stimulating, and then you get all of this output. Whereas here in this converging example, where all the input comes into one neuron, and then that's going to create some sort of an output. And that gets us back to like the summational. You know, if it's all in the same time and space, then that's going to kind of go back to this diagram, right? Here's our reverberating and our parallel after discharge circuits. So you can see here on the reverberating side that we've got some, it looks like it's creating a feedback loop, right? Because these neurons are feeding back into the loop, so it reverberates or resonates. It reminds me of like what we were talking about early on this semester about the negative feedback loops and things like that. And then we see here parallel after discharge. So you got all this input coming in, and they all come down to, that reminds me of, almost reminds me of the converging, right? Regeneration of nervous tissue and its repair on page 455. Although the nervous system exhibits plasticity, neurons have a limited ability to regenerate themselves. Plasticity is the capability to change based on experience, and then regenerate is the capability to replicate or repair. Neurogenesis in the CNS. So neurogenesis, genesis just means the beginning. So the beginning of neurons in the CNS is, in the CNS, there is little or no repair due to inhibitory influences from neuroglia, particularly oligodendrocytes. Remember we talked about oligodendrocytes at the beginning of the lecture. We'll go back and review those. Absence of growth stimulating cues that were present during fetal development, and then the rapid formation of scar tissue. So those can, in some cases, prevent neurogenesis in the central nervous system. Damage and repair in the central nervous system. The peripheral nervous system repair is possible if the cell body is intact, Schwann cells are functional, and scar tissue formation does not occur too rapidly. Steps involved in the repair process are, the slide should probably say damage and repair in the PNS, not the CNS. This is PNS, so this is another error on this slide here. This should be PNS. Steps involved in the repair of the process are chromatolysis, Wallerian degeneration, and the formation of the regeneration tube. Chromatolysis, the Nissl bodies break up into granular masses. This alteration is called chromatolysis. In Wallerian degeneration, degeneration of the distal portion of the axon and the myelin sheath is called degeneration of the distal portion of the axon and the myelin. So the part that's farther away from the center of the body. Formation of the regeneration tube is following chromatolysis. Signs of recovery in the cell body become evident. Macrophages phagocytize the debris. Synthesis of RNA and protein accelerates, which favors the rebuilding or regeneration of the axon. The Schwann cells on the other side of the injured site multiply by mitosis, grow inward toward, and form the regeneration tube across the injured area. And all that's on page 456 under damage and repair of the peripheral nervous system, PNS. So good point here that this slide should be damage and repair in the PNS, not the CNS. So this shows examples of those that we were just talking about. This is figure 1229 on page 456. Neural disorders, multiple sclerosis. So multiple sclerosis is a autoimmune disease that causes progressive destruction of the myelin sheath. Its cause is unclear, may be genetic or environmental. Symptoms include muscle weakness, abnormal sensations, and double vision. So I have a friend who had MS, multiple sclerosis. He had very hard time. So I remember going over and visiting him. He's really bad now. I haven't seen him for years. And as far as I know right now, he's bedridden, but he had a really hard time getting around. And I admired him because he lived on his own for a long time with this disease. And he claims that his cause was environmental. He got in a bad accident, and then after that accident developed the MS. I would say, but this disease, it probably could be genetic or environmental. It says, because your book, the slide says maybe genetic. I'm saying it could be either or. Kind of like scoliosis. We talked about scoliosis several chapters ago. We don't really know if it's genetic or it's environmental. Why does a young kid, a prepubescent person, develop this spinal curve? When the parents or there's no family history of it, or sometimes they develop that spinal curve when there is a family history of it. And it's the same thing with MS. That could be genetic or it could be environmental. And my friend thought that his was environmental. But you can live a long time with MS. It's just after you get to a certain point, you lose all of your ability to move. And I haven't heard from him lately on the social media. Sometimes I see his sister post something about him. But as far as I know, he's still alive, but he's not able to get around anymore. And he has to have people care for him 24/7. All of MS is disruption in the myelin sheath. So that is going to change the way that the propagation of the signal is moving through the axon. And then that causes, like him, his movements were real big, and he tried to fight it. I mean, he had all these exercise equipments, and he was doing treadmill and weightlifting every day, but it's unfortunate that happened. Depression is a neural disorder. There's several types of depression. Major depression, dysthymia, bipolar, seasonal affective disorder, or SAD. Common feelings are lack of interest in activities, sadness, helplessness, and possibly suicidal thoughts. Commonly treated through selective serotonin reuptake inhibitors. So there are some drugs that they can give you to help you with depression. And if you are depressed, if you're having these problems with your neural system, and you do have suicidal thoughts, there's always the suicide helpline. You can always reach out to somebody, and you can do it anonymously. So epilepsy is another neurological disorder. The short recurrent attacks of motor, sensory, or psychological function initiated by abnormal synchronous electrical discharges from the millions of neurons in the brain. So most people with epilepsy, occasionally, they might get some sort of stimuli where they go into a seizure. Excitotoxicity, destructions of the neurons through prolonged activation of excitatory synaptic transmission, and this is caused by high levels of glutamine in the central nervous system interstitial fluid. So that concludes chapter 12.